Hi, Cecile. Nice to see you.
Hi,
How are you?
Kat. Yeah, how are you? I miss you.
Me too. Sorry, I was. There's a. It's so annoying that while you're doing other things on the app, like add topics and all these things, you cannot control the microphone. Like it's so annoying.
Yeah,
Um,
it's hard. Yeah.
Hello?
Hello, Mike. How are you? Thanks for coming.
Excellent. Yeah, no problem. Good. I'm glad it's working. I was about to write you an email to see if I was in the right place.
Yeah. Let's wait a few minutes and we'll start um in like 4 to 5 minutes. Thank you.
Great. Yeah, great.
Hey, Katarina. This is Victoria. Are you
Hi,
there?
Victoria. Are you? Yep, I'm here. We're just waiting. A
Sure.
few. Yeah, we opened
Yes.
the room a little bit before to give people time to find the room, and then
Yes,
we'll
I
start.
hear. I heard. I was just wondering if you wanted to talk a little bit about the rooms that are coming up tomorrow as well, while we had people in here, or whatever.
Yeah, thank you. Usually I do it towards the end, but to not like confuse people. But thank you, Victoria, for reminding me. It's really good because sometimes I forget all of this. So yeah, thank you. Yeah. No, don't worry, Victoria. Thank you for reminding me. And hi, Eric. Um, okay, I think we can slowly start with introducing Mike to our club and the audience. So, yeah, welcome everyone to Science Society. We are very honored to have Dr. Michael Levine here. We had the room with him before with his colleague Josh Bongard. So some of you might know him. And yeah, we have a very like it's we are discussing a very interesting work of his. When I read it, I thought that this is one of the most important uh, preprints I read for this time we are in. So it's very broad and it goes into very interesting and breakthrough topics. So, yeah, I think this work, everyone that kind of thinks about the mind, the future, consciousness of the future, intelligence of the future, also medical developments of the future needs to read this. So that's why Cecile and we invited Michael back to talk about this because we thought it was such an important and amazing work. But first, let me introduce you to Mike for everyone that doesn't know him yet. Um, um, Dr. Michael Levine, he got his dual Bachelor of Science degrees in biology and computer science. And he did his PhD in Harvard in genetics. And then later he did a postdoc also in Harvard at in cell biology. And in 2000, he started his own lab at the Forces Institute, and he is now at Tufts University and Harvard Swiss Institute. His group works at the intersection of developmental biophysics, cognitive science, and computer science, and his mission is to understand how larger minds arise out of proto-cognitive subunits in diverse media. Yeah, he's a distinguished professor. He won a lot of different awards, and in 1995 he was chosen by the journal Nature as a his work as a milestone in developmental biology in the last century. So yeah, we are very honored to have you, Mike. We appreciate the time you make available for us. And yeah, the stage is yours.
Well, thank you so much. The very kind introduction. And pretty much, I'll just take questions. The only thing I want to say is that the preprint itself has has been now improved quite a bit over the last I don't know, month or so, and so there's a final version that's going to come out in one of the frontier journals. I'll put it up on my website, of course, and I'll tweet about it when it does come up. The final version's quite a bit better, I think, than the preprint. So check out that final version when it's there. But I think the main ideas are here. So yeah, if anybody is interested in specific parts, please let's chat about it.
Yeah, I don't know if you want to give like a short overview of what
Sure.
you
Okay.
wanted to address when you wrote it.
Okay.
If
Yeah.
not, we'll ask specific questions.
Yep, that's fine. I can give a broad overview. So, uh, the this is the second paper in a series, and there's actually the third one is being worked on at the moment. That started a few years ago at it was a Templeton Foundation conference in Scotland on diverse intelligences, and we were challenged to come up with a framework in which truly diverse intelligences could be compared with each other. And when I heard that, I thought that was an extremely important thing to do, because when I say truly diverse, I don't just mean humans, parrots, octopus, dogs, things like that. I mean truly diverse. In other words, given the bioengineering technologies, the possible exobiology, possible artificial AI, whether software or hardware. I'm talking about any possible agent. We ought to be able to have some kind of central invariant for what it means to be an intelligent agent that isn't tied to what you're made of or how you got here, meaning evolved versus design. I fundamentally take those things to be really contingent facts about something, not something that's deep. And I don't think there's any privileged substrate for cognition or intelligence. I mean, brains are not the only thing that that count as intelligent agents. And so I started, and so the first paper was in Frontiers, also in 2018, the Selves paper, and then this is next. And so what I tried to do is to come up with a few key ways to think about cognition, and in particular, this paper in particular defines my framework with the acronym TAME, TAME, which stands for Technological Approach to Mind Everywhere. The idea being that all of these ideas have to be empirically useful. That is, we can't just have philosophical sort of armchair preconceptions about what should and shouldn't be a cognitive system. We need to have views that drive experiments that enable novel capabilities. And so everything in this framework is all about developing empirically useful definitions of agency, of intelligence, of memory, of behavior, all of these things that expand rather than being restrictive. They're supposed to expand the kinds of experiments you can think of doing. Right? They're supposed to feed creativity and then be empirically testable. So that's the goal, and I can talk more specifically if people have questions about which kind of parts of it. We can talk about how I define all of these different terms. But all of it is sort of centered around two main ideas, and one of them is this idea of continuity. It's the emphasis on the fact that all of the interesting terms in this field, like intelligence, like cognition, and so on, are not binary categories. I think it's a really bad mistake to try to ask questions like, is something a cognitive agent, or is something intelligent, or does something have memories, or things like that, because that leads to all sorts of pseudo problems for reasons that I can describe. Um, that are not in fact that are not in fact addressing things in a deep way. But everything is a continuum, and the right questions for all of this is how much and what kind. And we can talk about that. And so that's the first thing, that there's this real continuity. And I think bioengineering now, chimeric technologies, bioengineering, and even aside from that, just an appreciation of ourselves as beings that are presumably cognitive, that used to be one cell. Each of us was just a single cell at some point, a collection of molecular pathways, and we all made this amazing journey across the Cartesian cut from just a collection of molecular pathways to a large being that has centralized perspective, preferences, goals, memories, and so on. So continuity, and then the idea of goal directedness. So this notion, and in my paper I formalize it and sort of make it semi-quantitative. This idea that you can gauge the intellectual sophistication of any agent, regardless of what it's made of or how it arose. You can gauge the sophistication of an agent by looking at what are the largest goal states that it's capable of pursuing. So in space and time, what are the largest states that it's capable of caring about? Another way, sort of an inverse way of looking at it, has to do with stress, and asking about what are the states of affairs? What are the biggest states of affairs that a system can possibly feel stress about? And and I think, if you tell me what you're stressed about, I can make a pretty good guess at what kind of on the scale of what I call a scale of persuadability, which is sort of a way to arrange different types of agents with respect to how much and how rich their cognitive lives are. We can kind of tell where you are on that scale based on what are the sorts of things that stress you out, all the way from very simple things like local sugar concentration, which will stress bacteria, to global of financial market states, and the well-being of people on a different continent, and so on. So very small states, very large states, and so on, and everything in between. So that I think is a kind of a 10,000-foot view of what I'm trying to do here. I'm trying to come up with a framework that really tries to pick out what is central and what is an invariant among all cognitive systems, as a way to help us relate to those systems. So understand them, predict them, in some cases control them, and in other cases communicate with them and relate to them in a kind of at a higher plane. So that's the introduction.
Yeah, thank you so much. I think it's a great introduction and overview. And I before if it's okay, I'll ask or I make a few comments. Actually, what I really liked about your paper is the very rational approach of the mind. To take this from a practical perspective. I think that is, what we need to do, and to also take the human mind off the pedestal, or even the mammalian network basically approach. That there are many ways to get there, and we need to have a more open minded approach to distinguish like diverse minds and intelligence. And what I thought it was also very interesting to think about the future, where let's say a human is 95% human and 5% something else, or maybe even just 5% human and the rest technology, basically. And that maybe with different rejuvenation techniques in the future, stem cells and so on, the amount of the original material basically decreases over time. And but in you describe basically that this is not a problem at all. Could you explain a little bit to the audience what you think defines the mind in this case, and how what mechanisms you know from your research that adapts to this transient modification over time of our organism, brain, mind?
Sure, sure. So I want to say a couple of things to that. The first is, let's just keep in mind, because I'm going to talk about all kinds of kind of unconventional intelligences and all kinds of weird agents. And what's important is that this is always done in a very engineering sense, meaning this is not just philosophy. It's not just empty sort of, I think that all the rocks are sitting around having hopes and dreams. The idea is that all of this, to the extent that it's true and useful, should be improving our technological capability. So, for example, when I treat cellular collectives during embryogenesis as a collective intelligence, it's because it allows us to achieve new things in regenerative medicine. It's because we figured out that you can actually communicate with the collective intelligence of cells with specific kinds of stimuli and reach outcomes that were not available to other approaches that treat them bottom up from as genetically controlled machines and so on. So all of everything I'm about to say is predicated on the idea that we will find out how much of this is true and useful by specific experiment. And a lot of these we've already done, but there's plenty more to do. So everything is designed to be empirically grounded and provide new ways to manipulate things like growth and form in, let's say, regenerative medicine contexts. Okay, so having said that, to get to the point of what what happens to when we really understand the future of bioengineering and chimerism. Uh, think about the fact that biology is incredibly interoperable, which means that I can take cells from different species, in fact, very widely, very diverse species of large evolutionary distance. I can combine them to be a new organism. I can include electrodes, various nanomaterials. You name it. You can combine things in really amazing kinds of ways, and the result of all of this is always some kind of coherent new living being. In other words, biology is very interoperable. And so what that means is that there's this really important tool called chimerism, so from making chimeras, making combinations of things, that you can deploy as a thought experiment first, but then actually at the bench, and we're doing some of that, and other people are doing some of that too, to really dissolve. There was a nice phrase, I forget who, maybe Dan Dennett or somebody used the phrase. It's a conceptual acid, and I think that's true. I think chimerism is a kind of conceptual acid because it dissolves all kinds of prior categories that have stuck around, not because they're true or useful, but just because of technological limitations. People couldn't really imagine how it could be otherwise. Uh, people and so I'll give you some examples. So here's an example. People often talk about the human brain. The human brain does this, the human brain does that, and usually this is in the context of saying, well, other things don't do that, and then they use this phrase, it's anthropomorphism, as if humans had some sort of magical powers, that it would be terribly wrong to think that these some version of these capacities exist outside. I always, whenever we argue about these things, I always point out to people that having a sharp binary category of what a human is completely unviable. And it's unviable for, I'll tell a technological story, and then I'll remind us that we already, even before the technology, we already should have known it's not viable. The technological story can go like this. It's very simple. We already have humans that have various implants that with various degrees of onboard intelligence that allows them to control modified bodies. So, for example, they run wheelchairs or assistive devices or prosthetic limbs that may have new degrees of freedom that they didn't have before. So basically, cyborgs, right? We already have people with all kinds of humans with all kinds of modifications that way. So imagine that you have a you have a house, and in this house there is a human that is sort of the brain is 95% standard human brain, and then 5% of that being is something new. It's prosthetics with some onboard AI perhaps that enable them to control devices, wheelchairs, vacuum cleaners, whatever it's going to be. And so this is not science fiction. This is already happening today. Next door, you have a slightly different. You have a different creature. What you have there is a Roomba. It's a computerized vacuum cleaner that is 95% vacuum cleaner, but on board it has some human neural cells in a little cultured dish that help the Roomba make decisions about where it's going to go and what it's going to do. This also is not science fiction. This exists. These are called hybrids, and people have already produced various types of artificial synthetic bodies for biological brains or, in fact, other cell types. So that already exists. So now you have 5% human cells in a 95% robotic body. So the important thing is that using these kind of technologies, or in fact bioengineering, we can produce any combination to any particular percentage breakdown. So if you want 70-30%, right, you want 80-20, any percentage can be created. And the combinations can be, and so I'm actually, this is another paper I'm working on right now on the space of possible beings, talking about how any combination of engineered material, evolved living material, and software can potentially be combined to be a new creature. So, what it is that you mean when you say a human brain, when it's very obvious that we can take steps away from the standard human brain and sort of make all kinds of in between creations with new types of capacities, new types of cognition. We could engineer a brain with three hemispheres. We could engineer a brain with cells from a human brain with cells of other species. All kinds of new architectures. All of this is now possible. And so any frameworks that we have for understanding what such creatures are can't possibly be based on some kind of firm natural kind, as if this was a a standard. This was a very clear cut thing that was obvious, where you could obviously tell where something is a human brain or it's not. There's an extremely smooth continuum, and not only that, it's a multidimensional continuum because you can alter it in lots of different capacities. Now, even before the engineering allowed us to do all these things, we should have already known, for example, just by taking evolution seriously, right? Pre evolutionary times, you could imagine a kind of Garden of Eden story where there was a discrete set of animals. Everybody knew what they were. You could number them. You could name them. There's a great painting I have that's called Adam names the animals in the Garden of Eden, and and that's it. There's a discrete set of animals, and we could say, okay, all these animals have some particular kind of primitive cognitive capacity, or maybe none. And then here are humans, and it's a discrete natural kind. But we don't live in that world anymore. We understand that there's a very smooth continuum of beings, all the way from modern humans, all the way back basically to single cell organisms, and before that. And so again, whatever it is that you think modern humans can do, you can track that back generation by generation. And even if you think something appeared quote unquote rapidly, where it's rapid maybe on geological timescales, but it's certainly slow in the biological scale. You will have every creature had parents, and going all the way back, and those parents eventually you track it down. You go all the way back to single cell organisms. So again, it's a totally smooth continuum, and The thing is that biology offers absolutely no place to draw a clean line to say that, okay, on the left side of this line are some parents, and these parents were not whatever it was, right? Name your favorite, cognitive, conscious, whatever you like. And then they had an offspring, and bam, on the other side of the offspring's on the other side of that line, and now you have true, whatever it is that people think of as true cognition, right? So biology offers no support for any kind of line like that, no support evolutionarily, no support developmentally. We all started life as a single cell. Gradually, we became this thing that now makes claims about its centralized cognitive states and its beliefs and so on. And so there's no line to be placed there, and there's no line that can be placed anywhere now that we understand chimerism and engineering. So that's my emphasis is on these kind of smooth transitions, which tell us that it's really impossible to operate in these tight binary categories as if they were some sort of natural kind, because they're not. Everything changes smoothly, and we need an invariant. We need a way to think about these things that is not tied to the frozen accidents of evolution, as far as how we got here.
What I also found was very promising based on your research and also on the paper that you write, that we don't need to know every single detail in order to come up with solutions how to, for example, in the future, treat different disorders. We just need to find the right triggers in order to make the system repair itself, or have some cells that come in and repair the system. Could you tell us a little bit about this?
Yes, that's right. And so one of the key concepts in this tame framework is this idea of the axis of persuadability. So let's just imagine it for a second, and then I'll tell you, I'll talk about how this relates to the biomedical side of things. The axis of persuadability asks us to, for any system that you come across, instead of having sort of preconceptions about what you think it should be, or how agential, intelligent, and so on it should be, to actually do experiments to find out where on that scale it is. And the scale has to do with what are the different types of tools that you can use to control the system. And control I use very broadly, meaning to get it to do something other than what it's doing now in a defined way. So on the left side of this continuum are things that basically are very simple machines, like clocks, for example. What defines these machines is the idea that you are not going to control them in any way other than through physical rewiring. So the hardware is your only option. So if you have a mechanical clock, you are not going to convince it of anything. You are not going to train it with rewards and punishments. You are not going to, reprogram it. If you want to control that device or interact with that device, your only choice is to understand all of the details of how it works. And if you want to build one, you have to specifically micromanage every part of what you think it's going to do from the parts. Okay, that's on the left side of the spectrum. Then you move on to something, for moving rightward. You reach the next step is very interesting. It's something like a thermostat. So the interesting thing about a thermostat, which are systems that can do homeostasis, right? They can do these loops, these homeostatic loops. What's interesting about those devices is that you can actually do better than rewiring, because you can recognize that they are working towards a specific set point, meaning that there's a state of affairs that's recorded somewhere as a memory of the set point. It's encoded in some kind of a physical medium, and you have an interesting possibility there. What you can do is you can control the system by changing the set point, by learning to read and write that set point, not by rewiring anything. So this is the way we control our thermostats, right? When you want the room to get warmer, you don't have to rewire the whole thing from scratch. You just go and you change what the set point is. You say, okay, now the temperature range that I want is higher, so go ahead. And notice something interesting there, that uh, by here's the trade-off: by recognizing that what you're dealing with is a goal-seeking system, you don't need to now know exactly how it works. The only thing you need to know is how to read and write the set point, And when you alter the set point, the rest of the system can be completely mysterious and it doesn't matter because you are no longer micromanaging it. You are relying on the fact that this thing has the competency to implement whatever goal state that you're going to put in. So that's pretty interesting. That's a new sort of great transition on the scale of persuadability. Then you move further, and you might encounter, and I'm only giving you sort of the main waypoints. There's lots of others in between, but then you can move further to creatures that actually have preferences, and what you can do there is apply rewards and punishments. Now this is even better from the perspective of control because you need to know even less about how it works. In fact, people have been training animals for thousands of, what, probably 10,000 years. They've been training, maybe longer, training animals, and the only thing you need to be able to do this is to know, a, to recognize that these creatures are in fact learning agents that have preferences, and b, to know something about the currency that motivates them. What do they like and what do they not like? What are the rewards and punishments, right? Once you know that, you don't need to know any neuroscience. You could train them and have zero clue about what's actually going on in their brains or anywhere else. In fact, you don't need to know that they have brains. You don't need to know where they store their memories. You don't need to know how they perform their homeostasis. You don't need to know any of that. You can motivate them and through behavior shaping and rewards and punishments. And then if you keep going further along this line, you may reach something like a human, where you have a next great transition where you can interact with those creatures with rational argument. So you're not directly rewarding and punishing them, you are giving them information and you are giving them logically structured reasons to do things. And you are taking you're offloading all of the hard work onto the fact that you believe they are to some extent rational agents, and they will alter their own behavior once they become convinced of whatever it is that you just told them. You don't need to provide rewards and punishments. They're going to handle even that themselves. So what you can see what's going on here as you move rightward on this continuum. The amount of stuff you need to know goes down. The amount of effort you need to put in goes down. The level of micromanagement you apply goes down. And what you gain is this ability to get more bang for your buck, so to speak. The ratio of how much effort you need to put in to communicate with this system versus what can come out of it. The complex, all the way on the right of the spectrum, one word that you might say to somebody that could have very low energy content event to say something to them could cause them to kickstart a massive amount of effort that they're going to then spearhead for years if they become convinced of what you're talking about, and you didn't have to micromanage it or put the energy in. So this is very important. So now let's get to the biomedicine part. The key part of this whole framework is that we should, when we encounter something, we should figure out where on the continuum it lands. So nowadays, molecular medicine, all of molecular medicine, is predicated on the assumption that everything is way on the left of that continuum. Everything is a simple machine. So what that means is that people will, of course, try to control it, meaning control medical outcomes, by manipulation of the lowest level components by rewiring the hardware. And thus, we have stem cell biology, we have genomic editing, we have rewiring of pathways, of protein signaling networks, all of this stuff. It's all hardware level stuff because people assume that that's the only way to interact with these systems, meaning that their intelligence and cognitive sophistication. I mean, they will say it's zero, but on my scale, whatever, it's at least very, very low. That's what everybody assumes. What we've been able to do in our group is by saying, well, you can't assume that. Let's actually find out where some of these things are. Then once you've said that, it actually opens. And so this is why I like these kind of frameworks, because they open. They're not restrictive. They open you to new possibilities and they help you do new experiments. So we've done new experiments. For example, we asked a simple question: uh, if you take a simple gene regulatory network model, which is just basically a bunch of genes that turn each other on and off in a network, everybody assumes that that's kind of a paradigm case of a fairly dumb machine. Well, it turns out that they can actually learn. They have 6 different kinds of learning capacity. Just already at the level of gene regulatory networks, you already have basic learning capacity. What we've also done is we've asked, what if uh, what if the ability of certain, let's say, animals to regenerate to after damage, or of the embryo to repair after damage, an early a mammalian embryo, you can divide them in, let's say, in half, and you get two perfectly normal monozygotic twins. There are many examples of this kind of incredible plasticity. So we said, what if all of this is a kind of homeostatic process? It's not just feed forward emergence of from of complexity from simple rules. I mean, of course, that also happens, but what if it's more than that? What if it's actually a process of remembering what the target morphology is supposed to be, what is the shape supposed to be, and then an error minimization scheme, just like a thermostat, that tries to reach it after damage. And if you do that, like all of the examples that I've given you, you can say, okay, I'm not going to rewire it. I'm going to leave the genetics alone. I'm not going to change the hardware. What I'm going to do is try to learn to read and write this set point, if there is one. And we looked for it and we found it. We found a bioelectrically encoded pattern memory that works as a kind of it's a sort of coarse grained descriptor of what the correct anatomy should be in a number of cases that we've looked at. And the system basically just tries to build whatever that says. And so what we've shown is that you can actually rewrite that information, not changing the machine at all, not changing the genome, not altering the cells in any way, but just rewrite that set point, that electrical information that the network is keeping. And sure enough, the cells will build other things. And when you do that, you can induce limb regeneration in animals that normally don't regenerate limbs. You can fix defects of the brain and face, the birth defects of the brain and face. You can produce two-headed flatworms and six-legged frogs and cause gut tissue to make a perfect eye. All of these things are possible, even though we don't know how to micromanage it. And I think for regenerative medicine, we don't want to micromanage it. I don't think in our lifetime we're going to know how to control all of the thousands of genes in the millions of cells that you need to control to, let's say, make a new eye for someone or make an arm. That's we're not going to be able to do that. We don't need to. The system already knows how to do it. What we need to do is learn how to motivate the cells to do it. So that's and this is just the beginning. I mean, the question, we are still not done asking about what else is possible, right? Where on this axis of cognitive capacity cells and tissues are. It may be that they're further to the right than we even thought, but for now, this is what we've been doing. We've been trying to understand how to uh, motivate cellular collectives to reach desired regions of anatomical option space for regenerative applications.
Yeah, Kat, may I ask a question?
Sure.
Yes, go ahead.
Yeah, Dr. Levine, yeah, of course, I've been following your work for more than 2 years now. And aside from morphogenesis, I'm a big fan of complex systems.
Mm.
So, but I'm really overwhelmed and I'm amazed at reading the paper and looking at the new work that you have, because I cannot catch up with the developments that's happening. It's happening so fast. And
Yep.
recently, I've read about your work on regenerating an amputated frog's limb, and yeah, I think you put it in an actuator and you place some cocktails on it and tried to let it grow back in about 18 months. Was that? Can you tell us a little more about it?
Sure, sure. Yeah, so what we are interested in the regenerative medicine side is really to identify triggers. We don't want to micromanage the process because, as I said, I don't think it's feasible to do. We want to find triggers or subroutine calls of specific complex patterning cascades. So in the frog, so frogs, unlike salamanders, frogs do not normally regenerate their legs as adults. So we took adult frogs, and this was work. There's a lot of people that contributed to this in my group, and also we did this together with David Kaplan's group, close collaborator. And I should, by the way, I should make a disclaimer from all this that David and I have a spin-off company called Morphoceuticals Inc. And so that's just kind of a disclaimer that I have to do when I give these talks. Um, and we wanted to do two things. Right after injury, we wanted to figure out a signal and then a delivery method for that signal that shifts the decision making of those cells. Instead of a scar, I want you to rebuild the organ that goes here. And so we found one. So David Kaplan's group made this thing we call the biodome. It's basically a wearable bioreactor. It fits onto the amputation wound, and then so their job is to make the dome, and then we figured out a cocktail of drugs that was supposed to kickstart the process and shift the decision of those cells towards limb growth. the coolest to me, the coolest thing about that study was this: the biodome was on that leg for 24 hours. That's it. So from the time we put from after amputation, we put on the biodome and we leave it there for 24 hours. Then we take it off. After that, we never touch the leg again. It that initial 24-hour signal, and maybe it could have even been shorter. We didn't try shorter. We just did the one trial, 24 hours. That 24 hours caused the legs to grow for 18 months, and they might have even kept going. We had to stop and and write up the paper. But 18 months of leg growth following just a 24-hour induction. So to me, that's very very important, because it uh, begins to validate this concept that what you can do is you can provide very brief triggers in that first 24 hours when those cells are deciding what to do. You shift that, and then you don't touch it again. It's not that we're going in and controlling stem cells directly or saying which tissues should be where, or anything like that. It's really a very early process, and then you get this massive amount of leg growth from a system that already knows how to grow legs. And yeah, and so that's what I think was interesting about that paper.
Just as a kind of an, I guess, tangent question to that, when looking at something like scar tissue, would it be feasible to say, let's say, look at some scar tissue and then remove it and then apply that same technique? Would you expect it to perform the same, regrow the limb, as it were?
Yeah, I think so. With one caveat, and we're currently, obviously exactly how it's going to play out in humans we don't know yet. But that's what in as a part of Morphoceuticals Inc. We're currently in experiments in a mammalian model. So we're now in mice with all of this, and hopefully eventually humans. Um, the thing with scar is this: I, bioelectrics in general, I think, from everything that we've seen, is a control modality at the organ level, not at the cell level. So people sometimes say, "Oh, can you use this to produce a specific cell type for, let's say, Parkinson's or for something else?" Maybe, but I think that's not really what bioelectrics is for natively. I think it's not for specifying individual cell types. It's for telling the cells what it is that they should be building. So if we have, so for wound healing or for skin repair or for clearing out unwanted extracellular matrix during a scar, in a scar, I'm not sure that bioelectrics is ideal for that. However, if you're dealing with a situation where you need to tell the cells to build a whole organ, it's not just a matter of specific cells. That's where I think the system is going to shine. So and now that's what it looks like now. I could be wrong, and so maybe at some point, scar repair is something that's doable. I'm not saying it's impossible. It's just that's not this is not about individual cell types or matrix or anything like that. It's about respecifying what the cells are going to build.
Yeah, because I'm just thinking of like people who have burns or other injuries. They've already had scars grow over the area, and
Yeah,
if you removed that scar tissue, that could certainly improve the quality of life if you were able to kind
yeah,
of regenerate their various tissues to the way that they were originally. So that's very exciting. It's very much Star Trek. So hell yeah.
yeah, 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 yeah.
Hi. Hi, Michael.
Hi.
It's great seeing you again. I think that we talked about one of your the Xenobot projects that you were doing,
Mm-hmm.
and it's really great to see another perspective, which is also as illuminating and thought provoking. Had one question, which is you mentioned something about the sort of like the consistency and coherence from single cell all the way up to like a multicellular organization such as us, that we do have something in common when it comes to solving particular problems or addressing the particular problems. And you also talked about the different mechanisms which are currently been employed in tackling those problems, which is sort of like removing a single gene at a time, and probably investigating um, the the physiological response, or the uh, essentially, like, the change that the organism will go through is not probably a very a good paradigm. And I think most people in neuroscience, because that's probably the area that I feel comfortable in, that also recognize this, that knocking out some genes is not probably going to solve everything. You're just going to make a crude assumption as to what the behavioral and other responses might be based on just simply omitting one single gene. But my question to you is that for complex networks, like what is your recommendation in order to kind of error correct that? And by that I mean, like, for example, we do have like um, like for human brains, for example, right? If you have lack of dopamine surplus or a dopamine supply, that you get Parkinson's disease or something like that, which is sort of like working on a network level as opposed to, or at least like cascading into like a network level as opposed to just a single cell issue. But what do you think like is the best way to kind of address when a network is not behaving the way they expected? And by network I mean could be multicellular organization.
Yeah, I think that, and of course, everything I say is not meant to suggest that this is the only way of doing it or that we know everything about it or what to do. In fact, we don't. I think this is just the beginning of, we know hardly anything about most of the important questions. But the strategy that I always prefer, as much as possible, I try to ask what is the highest level at which I can control this. So if there is a way to do it via training, if there's a way to do it via some things. I mean, neuroscientists are actually really really good at this because they're comfortable with the idea that there are multiple levels of organization. So for example, you've got people in neuroscience studying specific molecules and synapses, somebody else is studying network activity, and somebody else is doing psychotherapy, right? All of these are ways to manipulate the system at different levels, and different problems are best addressed at different levels. Sometimes sometimes the best way to do it is to adjust the levels of a particular neurotransmitter. That's fine. Other times, you want to do some kind of cognitive behavioral therapy where you're literally going to train people into specific behaviors. Other times, somebody, there are many different ways. If you think about, for example, depression, right? Some people, the cause might be literally a neurotransmitter imbalance. Somebody else might be in that state because they're a deep thinker, and various skeptical arguments have convinced them that they're just it's just they're kind of in a philosophical deep hole where they find it hard to motivate themselves to do anything given that the universe is going to burn out. So you're not going to treat that with a neurotransmitter adjustment. So in all of these cases, you want to find the optimal level of control. And I think for medical purposes, the higher you can go, the better, because then you're much more likely to produce a good effect, as opposed to trying to micromanage the thousands or millions of different components that you have to be in charge of when you're addressing it at a lower level. That's how you avoid all of the side effects. I mean, this is a whole other thing we could talk about, but um, all of the side effects of modern drugs, and the way that pharmaceuticals are used nowadays is basically at the lowest level to try to treat symptoms, right? We have very few, if any, other than antibiotics, we have very few, if any, drugs that actually fix anything. They're mostly designed to keep the symptoms down while you're taking them, and as a result, of course, other stuff crops up. You get all these crazy side effects because you're trying to micromanage everything yourself, and it's extremely difficult. The future of all of this is understanding the learning capacity, understanding the motivations of all the different subsystems that you're working with, whether it's kidneys, livers, brain tissue, it doesn't matter, and trying to push them into health states by as far up as you can, as opposed to trying to manage the individual outcomes.
I had a question, and it comes from an assertion that I've had from some time, but I need to just put it into some context. What I find fascinating about your approach, particularly the continuum from complexity across from multicellular organisms right up to humans. In the sense of uh, general AI applications,
Mm.
which I often end up evaluating in industries where it has a very decided engineering context in the defense industry, for example. Um, the assertion is that you don't get general AI without a body. And it seems from your work you might agree with that. But it there's a very practical engineering sense that if control systems that are going to make decisions rapidly, um, if they are going to need to abstract and as I thought your construction of the axis of persuadability was brilliant. But if we're going to get control systems engineered to be more to the right on that spectrum, is there a need, or would you agree, or at least reflect on the assertion that it has to be coupled into a physical system, in the sense that the t- timing matters, coupling into having an actual physical sensory control loop is
Yeah.
critical. And I say that even in the sense that taking computational algorithms into hardware in the loop scenarios, it's still often a leap because any thread can run out of time and you're up against a hard clock and you just don't get that time. But I think it might be deeper than that, because even with real-time systems, you can remember where you were and pick up. You can't be so violently as disturbed as to forget what you were doing and have to recover. Um, so I just thought, in that sense, the assertion is you don't really get general mind without a body. What would you say to that?
Yeah, very, very, very good, very important point. Okay, here's what I think. I think that's true that you do in fact need a body. However, I want to redefine what a body is, and I want to let go of the assumption that it's a physical body. And what I mean by that is that in fact, none of us really know that we have a body, right? This is an old kind of a very old idea, and we've now seen that biology is extremely modular. People have taken cells in a petri dish and connected them with electrodes to a flight simulator game on an Xbox. And then you sort of, every time the plane crashes, you shock the cells. And guess what they learn to do over time? They learn to keep the plane afloat. And so this kind of thing, the loop is there. I think the loop is absolutely critical. I think some constraints on that loop are very important to motivate the agent. So you have to do this correctly. It's not sort of any old virtual setup will do. However, I think that we are all operating in virtual worlds, and this is very close to the ideas of Don Hoffman, for example, where none of us interface to the external world directly. Our perceptual systems don't tell us actually what's out there. All we get is various kinds of user illusions that are helpful for us to manage our sensory inputs. And so uh, I view intelligence as competency in navigating space, but it doesn't have to be three-dimensional space. So, for example, cells will navigate metabolic space. They will navigate physiological space. They will navigate morphospace or anatomical space, right? The configuration, all the possible configurations of your body. Um, in fact, we even have robots now that navigate these various spaces. For example, a smart insulin pump is really a robot, a Bratenberg vehicle that drives around in physiological space because it measures various states and it applies corrections, and it has states it likes and states it doesn't like. So all of these things have a degree of intelligence that operate in all kinds of spaces. I mean, cells can operate in the extremely high dimensional gene expression space, right? The space of all possible gene expressions. And they find, they navigate it with various degrees of competency, meaning that under novel perturbations and in novel environments, they still can get to where they're going. And of course, sometimes they screw up and so on. All of these things are intelligence, but it doesn't have to be three-dimensional space. And I think two important things to say about that. One is that evolution makes use of this extensively. So we started out as protists that basically could only handle a metabolic space, and then evolution pivoted those same strategies, things like run and tumble and so on, pivoted those same strategies to then handle other spaces, for example, transcriptional space, developmental, anatomical space, and eventually, when brains and muscles evolve, then and only then you get the ability to navigate three-dimensional space by moving around. By the time that arose, and the kind of environment that we think of as behavior, right, the three-dimensional environment, by the time we were able to move around in there, evolution was already old hat at solving general intelligence problems just in other spaces for creatures that physically don't obviously move or do anything. Now, the other important thing to think about is this: when we look in the world and we see something and we try to take a guess as to how much intelligence it has, what kind of problems it's solving, what space is it working in, we are basically taking an IQ test ourselves, because our estimate of this is only as smart as is only as good as our own ability to recognize this, and our ability to recognize that stuff is really bad. Um, because we grew up as individuals and as a species with all of our sense organs pointing outward into the three-dimensional space. We're really good at detecting agency in the three-dimensional world. We all know the difference between a bowling ball and a mouse, and all animals immediately have this intuitive feel for what they can expect from various objects in the world, because they're used to moving things moving around and dealing with that. Imagine if we grew up from our earliest days with a biofeedback sense that told you what your pancreas was doing at any given moment, right? Imagine if the way you have eyesight, you also had another sense which had a rich data stream on what was happening to your bloodstream and what your pancreas was doing about it. I think if we had senses like that, we would have no problem recognizing intelligence in physiological spaces. We would know that this is an intelligent object, that it has particular things it's capable of, other things that fool it. We would know ways to ways to help it along. We would know ways to trick it. All the things we do with conventional intelligences, we are just not good at recognizing them in unconventional spaces. Same thing for transcription. We look at cells. I mean, I could tell you specific examples of cells doing amazing things to figure out what genes that they should be turning on and off, and solving novel problems. That kind of general AI is already here, and it absolutely requires a body, but not the kind of body that we think of. And so I think, based on the work on hybrids and instrumentized neurons, they play Pong, they do all kinds of things. Based on based on all of that, I think that biology does not care what kind of space it's working on. It's very good at porting intelligence between spaces, and I don't think it cares whether this space is quote unquote physical, meaning it's a three-dimensional space, or it's a virtual space, such as maybe a kind of a VR setup, or a brain in a vat kind of situation, or a transcriptional space, or any of this other stuff. So I think that's my sort of long-winded answer to that. I think yes, you need a body, but bodies are not what we think they are.
That's interesting. That kind of reinforces a recent paradigm shift where the microbiome itself is sometimes seen as another sensory organ, and the kind of information that we're getting from it could perhaps be harnessed and better understood. So this seems to be going in that direction. That's very exciting. I'm curious if you think that there's, because you said it was an electrochemical reaction. Do you think there would be any possibility for any sort of electromagnetic interaction, or have you looked at that in any way?
Um, I mean, basically, the thing with bioelectricity is not some magical medium that's required for intelligence. What's important about bioelectricity is that it's a very convenient piece of physics with which to do feedback loops, memory integration across space. I mean, evolution uses it all over the place, in brains, the rest of your body. We use it for our computers. It has useful properties. And so I focus on the bioelectricity because it's a great way to illustrate some of the points I'm trying to make. I'm sure there are other media. I'm sure you could, in theory, do the same things with electromagnetics. I certainly have not. I don't think anybody has. I'm not saying it's impossible. I'm not saying there's not some giant cloud somewhere in the universe that uses magnetic phenomena for the same to be intelligent the same way that we use electrical synapses between cells to scale up our goal-directed activities. I'm sure it's possible, but no, we've not done anything like that. I don't think anyone has yet.
Yeah, I think the closest thing was Lee Cronin talking about potential molten creatures that use or harness electromagnetic fields for that kind of computation, but that was
Sure.
very exotic, kind of
Yeah.
far out there little promotional
Yeah.
science video that I've seen. But
I don't
thank
see
you.
why not. I don't see why not. All I know is that we are really bad at identifying agency in unfamiliar guises to the point where people, especially scientists, are willing to make armchair pronouncements about what can and can't be an agent, and we really need to get better at it.
Very fascinating. Yes, Willie speaking. If I may chime in, I have three questions, but I came a bit late to the room, so I'm sorry if you already made some statement and my questions are redundant. The first is about the status, the second about maturity, and the last about kind of epistemological question. Uh, if I get it right, the backstory is you were researching this Xenobot thing, and actually you are now, as a side effect, have a new paradigm. Eric just also used the term paradigm, uh,
I mean,
maybe
that's
in therapy, in medicine. The-
so temporally, that's not how it happened. This the ideas around this paradigm, I've been working on this for uh, probably 20 years or so, and I've been sort of very slowly rolling them out for various ways we can discuss about the sociology of science. But the Xenobots have been a very recent kind of a very recent invention that certainly helps illustrate these points and drives some new aspects of the framework. But this was not created by the discovery of the Xenobots. No, this predates that
okay.
by many years.
Okay, okay. And of course, you stand on the shoulders of all this. Uh, I think you even mentioned René Thom or morphogenesis and other kinds of system theory. But what I've had the impression, but I did not read your paper, sorry for that, so far, that because you did not mention cell communication, maybe you're leaving this as a too simplified paradigm or framework, for instance. This is one question. And
So
what I meant
yeah,
with
so
paradigm is that it sounded like you have found, oh, sorry, I'm in the kitchen, you discovered a new kind of interaction between also say the doctor and the patient, because we were talking about these trigger points and calling the subroutines, and this is a totally new paradigm to do medicine if it's this what I've got. And this was about the first question about the paradigmatic status, which could be for medicine. That's what I mentioned, not in the research program you are doing.
sure. So yeah, feel free to connect with me offline, and I will send you a bunch of pointers to work. So first of all, the cell to cell communication, yeah, it's absolutely critical. We've been working on it for 2 decades now. It's absolutely critical. We have lots of work on it, and I think you're right. I think there is a new way to to look at this as a new mode of interaction between medical interventions and the patient. We have lots of work on this in very practical, in regenerative medicine, in cancer
Mm,
remodeling, all of this stuff. So yeah,
mm.
yeah, I think
Yeah.
you're right.
Okay. And what you talked about, the micromanagement as a as an opposite thing. Like if you found the right points, and this reminds me even, sorry for that, to the trigger points in some more alternative medicine space like acupuncture. So maybe it is related in a way that there was some discovery related. I don't know. I don't want to discuss now. But the other thing is, which is the maturity status of this? Is it like you are in a more alchemistic state here, that you are experimenting and don't really know what will happen next? This is also super valuable because this is the way forward in science, to really keep on track without knowing what will happen, just experiment. or are
Right.
you
No,
already in the
that's
stage to have a kind of periodic system, what you do with the cells and what you do with this transmitters or whatever signal messenger stuff or like this? Yeah.
yeah, yeah. As of the last 6 or 7 years, we've been in the latter stage. Basically, we are now to the point where we have computational models that tell us exactly what to do for specific cases. Now, I will say that all of this is at a very early stage because we can only do this in a very limited number of cases. But I'll just give you a very kind of simple example of what we did. So. If you look at what controls the development of the early brain in the let's say in the frog tadpole, the early brain, which is a very complex organ, it needs to know where it should form and how big it should be and what its structure is. Okay, and we found out that there are a variety of birth defects that can be induced by either mutation or exposure to teratogens, let's say alcohol, nicotine, many different things that screw up this ability, and they have terrible brain defects, the no behavior, and so on. What we were able to do was make a very specific computational model of what goes wrong with the bioelectrical signaling between the cells that causes them to not be able to remember what the size and shape of the brain is supposed to be. And having created that model, we were able to ask them to basically invert the model and ask it a question, which is which ion channel proteins would we need to open and close to get back to the correct electrical performance of that network? And the model was able to suggest a specific channel that it predicted would give back the proper patterning behavior of that electrical network. Once you know the channel, you can go to your shelf and pick out a specific drug that opens that channel. And we did it, and sure enough, everything became correct. So the brain structure was repaired, the brain function was repaired, the animals got their IQs back, meaning they could learn at rates indistinguishable from controls. So what you have here, and this was 2018, so this was like 4 years ago. So what this shows is that at least in some cases, we are able to make a very specific computer model that tells you exactly what's wrong, and you can invert it to look for interventions and make predictions about exactly how to fix it. Now, like I said, this is we are only able to do this in a few specific cases. So we've done some tumor reprogramming, we've done brain and face repair, we've done limb regeneration, a couple of other things we can talk about. We do not have this in the most general case. I mean, sometime in the future, you will have what what we're working on, and hopefully I'll someday I'll get to see it. Uh, this this platform where you basically can pick any organ in the body and and get a prediction about what bioelectrical state needs to be induced to repair that, and then you can pick an existing drug, and there are an enormous number of what I call electroceuticals, which are ion channel drugs that are already approved for human use. And the computational model will tell you which one. Someday it will be universal. Right now, we can only do it for a small number of indications, but it's very important to understand that this is not random poking around. We are not just doing random
Mm.
perturbations to see what happens. That's how we started in 97, 98. That's what I was doing. But we've gone very far since then, and at this point, at least in some of these cases, it's very much rational control. It's not random
Mhm.
discovery.
What pops up in my mind is it's a kind of interventional framework, a new type of interventional framework. How do you call it, or do you have a name for this concept, that general concept you are doing now? You're
Yes.
applying?
Yeah. So the framework as a whole is called it's called TAME
Ah,
for
yeah, yeah, of course. Yeah, that's what I
technological
saw.
approach to
Mm.
mind everywhere. Now
Yeah.
we have in the now you got to realize when I publish papers in regenerative biology and medicine journals, and when I talk to people for our spin-off companies and so on, I don't talk about any of this stuff. These are things that the majority of people who are really into molecular medicine and so on, they really don't want to hear about any of this. And so
Mhm.
I have a different a different lens on all the stuff that I can talk about when we're and now when I started all this, I didn't talk about any of this at all. Everything was well, that's a whole other discussion about those kind of
Mm.
sociology of science.
Mm.
But what really drives all of these approaches is really keeping in mind what kind of agent are we dealing with? What is its intellectual sophistication? What are the motivating trigger points? What does it know how to do? What are the preferences? That's the sort of approach. But of course, uh, this is certain
Mhm.
audiences are interested in that and others are not.
Yeah, sure, but this will change soon, I guess, because I think it is a new interventional framework how to interact with organisms in general, or tissue and so on. And so this is why I think this room could be, I'm not kidding, historic, because it's introducing a paradigm. Of course, you're doing this for years, I know, but this is kind of a public space. And the last question now is about the metaphor or the modeling or the epistemology, because what popped also up in my mind was a kind of a metaphor of the conductor of an orchestra. If you think about that an orchestra could not really be able to play a symphony, so to speak, if the conductor is 500 meters away, then it's like we've done medicine before, and in the future maybe we just found a way to have this little stick for the conductor, and people in this way, the musicians are the cells, are listening in the right way, and you know what to do with your stick. And then of course it's very efficient, because as you already said, like you have low energy to introduce, for instance, to persuade a person on a rational basis. And in the same way, you can, if you find the right trigger points and so on, um, persuade, so to speak, some cells to rejuvenate or something like this. So, do you think, because you had this acronym, um, mind everywhere, yeah, it's like, of course, not esoterical way or metaphysical way, but it's just next stage of systems theory that you found something like, yeah, similar bit to attractor nets or something like this, which are like a spirit, or yeah, and I avoid this metaphysical term, but exactly yesterday, Joscha Bach was on a Zoom call, and there was also von Glasersfeld, the old guy. You probably know him from at least from literature, and they were talking about attractor nets, just to connect this. And maybe it's interesting to see that. What you call a mind is a metaphor for what's going on in the living systems, whether they are chimera, or whether they are very basic tissues, or they are cancer, or whatsoever. That's an interesting thing. But maybe we need a new metaphor that people don't think they talking too metaphysical. So thank you for your brilliant answers. Thank you.
Thank you so much. Yeah, thank you for your comments.
Yeah. So I have a one comment. I just wanted to know what do you think about this. So I was reading this very famous book um, from Francisco Varela, like Principles of Biological Autonomy,
Yep.
and there he defines kind of like autonomous systems and control systems. And for the control systems, he says that the fundamental paradigm of interaction is instruction, and the unsatisfactory results are errors. And for the autonomous system, he says that the fundamental paradigm of interaction is a conversation, and a unsatisfactory result is are breaches of understanding. So I found that what you said from the beginning, that like people in molecular biology were trying to do this kind of from the like let's say the smaller components, kind of try to do an input and output, um, let's say, process, a sort of control process. But as far as I understand, what you've realized, what you've discovered, is that this bioelectricity works very much like a conversation with the cells, and you are like having a conversation using all these cognitive concepts, kind of translating them to bioelectricity concepts. Is it more or less correct?
Yeah, I mean, one of the things you can do, because the basic fact is that everything that brain cells do, so neurons, everything that neurons do, was not invented fresh when neurons appeared in evolution. All of this is extremely old. So the brain basically co-opted all of its tricks from much earlier cells that were already using electricity to communicate in networks. It's just that they were using it to solve problems in anatomical space instead of in three-dimensional space by movement. And so that means that what you can often do is take any neuroscience paper, put it into Microsoft Word, and do a find replace. And you can anywhere that it says neuron, you replace that with the word cell, and anywhere that it says millisecond, you replace that with hour. And once you've done that, you get a very interesting paper full of ideas for developmental biology experiments, because the way that these things work are very parallel. So what we have in cellular collectives is a kind of collective intelligence. I mean, so are we. We are all, in fact, all intelligences are a collective intelligence, right? We are all made of parts. So we happen, when you think about yourself psychologically, you're referring to a collection of largely neurons, but there are other sort of nonverbal entities that are made up of collections of other cell types. And I completely agree that in the end, a proper kind of an optimized control policy is going to look very much like a conversation with these cells. Now, not with individual cells, by the way, with cell groups, with cellular collectives. The same way that you and I are not having a conversation between our individual neurons, we're having a conversation with the whole system. So yeah, I think in the end that's probably what it's going to look like, but it's unclear exactly where on that continuum these various, let's say, developing embryos, tissues, whatever you're interested in, where those things exactly lie. This is where experiments have to happen. We cannot assume anything. We have to do experiments and see which types of interventions, be they conversations, be they rewards and punishments, so in other words, training protocols, be they physical rewiring, which and the many others we can talk about, which of these things is going to be the most efficient way to relate to that system. I mean, the important thing is this: a lot of people, especially in science, are terrified of making a mistake in one direction. That is, um, I call this a teleophobia. There's sort of it's terrible to ascribe too much cognition to some system, and then your colleagues make fun of you and all of that. But of course, going in the other direction is just as bad. If you don't recognize how much intelligence a particular system has, you are going to be in terrible shape when you try to relate to it. You're going to be wasting your time and leaving all kinds of capabilities on the table if you are trying to relate to a complex intelligent system as if it were a clockwork, right? There are going to be so many things that you are never going to do, and many other things that will be too difficult for you to achieve. So making a mistake in both directions is bad. We need to do our best to gauge the correct level of interaction, and that might turn out to be conversations, and it might turn out to be training, and who knows, it might turn out to be other things we don't know yet.
I wondered if I could just point out that it may be even older than that. So there was a biologist at my university, UBC, I can't remember her name, but she argues that the microrhizome structures also have
Mm,
the same distinction between
mm.
conversations and instructions, and that under the ground, unbeknownst to most of us, are all these kinds of complex interactions that we tend to think of as originating in animals and in organisms, but in fact they're much older.
Yeah. No, absolutely. And these there are things that are happening in bacteria, and yeah. All of these things are experimental fodder for asking what is the maximum level of sophistication I can take advantage of. And in some cases it'll be high, and in some cases it will be low. But it has to be discovered empirically.
So thank you so much, Michael. That was very fascinating to me.
Thank
So
you.
I do have a question. Do you think that your model, because I'm seeing here that you talk about synaptic vesicles, which you introduce as an exosome, and you have some description about the volume transmission. So I was just wondering, do you think that your model have a capacity for memory storing between the connection? I mean, I mean,
Uh,
in the form of the
I,
connection between the cells, actually.
I mean, so I'm not sure if we're talking about the same thing. I don't recall actually saying much about really vesicles,
Yeah,
but
that was you just posted
where
on the top. I mean,
I'm sorry, I don't know where I'm not sure where we're looking, but for sure, I think memory, yeah, absolutely. So cells connect. I mean, mostly what we work on is gap junctional connections, right? So we work on synapses made of direct electrical connectivity between cells, and for sure, there's the ability to form memory there. In the collectives, right, in cellular multiple cell groups connected by gap junctions, absolutely can store memories, and we've worked on that a lot. Vesicles, I know almost nothing about vesicles, but there may be similar capacities there.
okay, thank you so much. I just found it from the, document that you just posted on the top. That was in one of the pages. I just pay attention, and that's why I just came up into this question. Thank you so much,
Yeah,
anyway.
yeah. No, drop me an email it's offline, and we can take a look at it and see what we're talking about.
Sure.
Yeah, if I may just there's like a release probability differences between different inputs and synapses, and they can change with excitability changes and memory formation, like long-term memory formation. So, if I would pinpoint it to something, I would say, there's like different there's like a malleable way to change release probability of vesicles and then neurotransmitters and things like that. And then also the uptake of neurotransmitters would probably be another way of changing and adapting.
Mm. Yeah, and that sounds perfectly reasonable. I'm entirely possible, but we also have to keep in mind that there are many systems that can do learning and memory that are single cell or that have no synapses at all. I think the notion of synapse is pretty critical. But what's important about it is not the type of synapse that we specifically identify in neurons. What's critical about being a synapse is that it has historicity. It's a it's the action its state right now is a function of what happened previously, and that you can make out of transistors do that, different kinds of materials do that. There are many ways of making something that functions as a synapse, right? And neurons have one version of this, but there are also many others, and that's gap junctions are the ancient version of that. uh, so are ion channels for that matter. Lots of different ways to make that. In fact, at one point I was toying with this idea of trying to design a gravitational synapse. Right, you can imagine this enormous kind of planet, solar system scale kind of thing, where there's a particular state in the distribution of its orbiting bodies where signals come across to it, meaning some kind of mass will come flying by, and that permanently alters the pattern, so that the next time it comes flying by, something different will happen because the state has changed. So you can imagine building these synapses out of very diverse materials at different scales, both temporal and spatial. Yeah, there's lots of ways to make synapses.
Yeah, I love that about your work, that it doesn't restrain us to anything and like to any type of mind or, whatever we want to call it. So I really love that idea. And it's funny that you're saying that on a planetary level. I read an article yesterday about astrobiologist talking about the planetary intelligence. It's funny that
Mm.
it's a nice coincidence that you mentioned that.
Yeah, and it's one of the things that I often think about is um, how much knowledge can we have if we know that we are composed of parts, and presumably we are also part of larger systems which themselves may or may not be agents. That again, experimentally, you'd have to find out. But if we are, how would you know? So for example, if the solar system was in some way embedded in this giant gravitational network structure that was propagating information, could we tell? And at a slightly smaller scale, if we as humans were part of some kind of functional larger network, some kind of society network, could we tell that in fact that was happening, and that it had goals in the same sense that we have, that is probably invisible to ourselves, and the very and our and organs and so on. So I don't know if we have the mathematical tools to even, or if that's the sort of thing that's forever undecidable. But if we have the tools to be able to recognize that you are part of a greater system that is an agent, in the same way that we know we have components that are part of us, that don't have access to all of the cognitive information that we have as a composite system.
Yeah, that's interesting. I was shortly while I was driving in Professor Lecormand's room about what is life and new ways of thinking about that. And it's very interesting that you and his approach come from different backgrounds, but basically, uh, I think, go in the same way. The main idea was that we don't necessarily know what life means because we have a way too narrow way of thinking about it and seeing it, and that's why we didn't discover life around the universe so far. So I think to have this very broad mindset will enable us to see more and discover more in the future. So yeah, thank you for that.
Yeah, thank you, that's great.
Jonathan, you came up to the stage, but before that you speak, I wanted to ask Mike, how much time do you have left? I didn't want to stretch your patience too
Yeah,
much.
yeah. I've got 8 minutes before I need to go make the kids dinner. So 8 minutes.
Okay, great, thank you. Okay, Jonathan, go ahead with your question.
Sorry, I don't have a question. I just came just back in the room and I was confused that I was suddenly on stage, so apologies.
Oh, no problem at all. Yeah, if anyone has a last question, yeah, Eric, go ahead.
Yeah, hi. So I guess another question is uh, the complex relationships that all these systems have. Um, were there signals that kind of determined the health of a system in the sense of like a pathogen? So were there ways, or is there any work going in that direction? Just curious a little bit, just because we're in the age of COVID and everyone's focused on this, and it would be really cool if we could just tell our body specifically what to do, since we seem to know what our body should do, but we just can't seem to communicate that to the cells themselves. So is there any future work perhaps in that direction?
Yeah. We don't specific. I don't have anything specifically, sort of exciting about that in terms of a pathogen infection. The one thing I can say is that what we have discovered is that information about defects of all kinds, and I think probably pathogen infection will end up being one of them, although I don't know that for a fact, is information that's available throughout the body. So, for example, if in a frog, if a frog loses one leg, let's say at the knee or some other position, the opposite untouched leg that nothing has happened to it will light up with a bioelectric signal in exactly the same location where the opposite leg got injured. So within 30 seconds, the opposite leg finds out within 30 seconds what's going on the other side. So that opens the way to what I call surrogate site diagnostics, where you can look at one area and find out what's going on in some other area. I have a feeling that that's just the beginning. That it's probably a very rich area. Harold Burr, back in like 1938 or so, was publishing papers on using a voltmeter on the skin of a rabbit to detect tumors that were, I think, in the cervix, if I recall correctly. So again, long range detection of disturbance of morphogenetic order. So yeah, one thing these bioelectrical networks are very good at is propagating information across distance. And we've certainly had both in cancer and in repair of birth defects shown that you can fix things at one location by doing manipulations at a completely different location. So I think all of that will have a huge future, but the infection is not something we've tackled yet.
Okay, well, that leaves some work for the rest of us since you seem to have done so much already. So,
Well, thanks. Yeah.
Okay, so I think, yeah, we should thank you. Like, thank you very much for coming and presenting your very groundbreaking research. I
And thank you
think,
very much. Yeah, thank you for having me, and
yeah,
thank you for all the great questions. Some very, very nice discussion questions. Thank you all.
yeah, and come back anytime as usual. And yeah, again, I can't thank you enough. I think your research is groundbreaking, amazing, and everyone here probably wants to be you when they grow up. So thank you so
Well,
much.
thank you so much. Yeah, that's very kind, and I really appreciate the interest and the support. And yeah, be well, everybody.
Thank you.
Thanks.
Thank
Thank
you.
you.
Thank you.
Thank you.
Bye.
Thank you, Michael.
Thanks very much. Thanks all.
Just shortly to make a couple of announcements. Tonight at 10 p.m. EST, we have Dr. Kaufer. She will talk about her research about brain changes in the brain and anxiety and PTSD state. She was in the news everywhere with her groundbreaking research. So yeah, join us at 10 p.m. EST. She just joined Clubhouse for us, so yeah, it will be a great talk. She's a very interesting person, also nice to talk to. And then we will have tomorrow another room, and it's a computational approach to study pathogens. The researcher from Berkeley, she uses machine learning to study pathogen evolution and also different interactions with the host. So it's really important work, especially now in sight of having a pandemic, and it will hopefully give us a lot of insight for the future to protect us from pathogens. So yeah, and she is very well prepared, and yeah, she's also a very great researcher to talk with. And yeah, please come back. We have many more rooms. Join Science Society if you like this type of discussion and learning from real scientists with real publications and peer-reviewed journals. And yeah, thank you everyone for coming. Thank you so much, Cecile, Eric, Abyss, Willie, BA, Lian, Parkash, Karan, and everyone in the audience. I really appreciate it. And see you back in a few hours, in 4 hours and a half. So I'm looking forward to it. Thank you, everyone.
Thank you, Katerina.
Thank you.
Thank
Thanks,
you.
Kat.